Hello students, good morning. I'm your educator Dolce and today we will start with the third phase of the M phase of the mitotic division. So what we have done in last lecture, we have done the prophase in which the condensation of DNA occurs. Next is the metaphase in which the chromosome, the condensed chromosome, they align themselves at the equator, that is said to be the metaphasic plate. The metaphasic plate, it represents a type of arrangement of chromosome at which the spindle fibers or the microtubules get associated with the kinetochores around the centromere, so that in the anaphase the chromosome get pulled via shortening of the spindle fibers to the opposite ends of the poles with equal frequency. So what happens at the onset of anaphase, each chromosome get arranged at the which is arranged at a metaphase, it start to split, means it start to break or to separate from the chromatid part, and the two daughter chromatids, now referred to as a chromosome, they move towards the opposite pole, that is said to the migration begins across the opposite poles. As each chromosome moves from the equatorial plate to the opposite end, so the chromosomes start to separate, and this leads to a path in which this kind of figures appears. As is shown in this figure, in the anaphase the chromosome get condensed or they move toward the opposite pole via shortening of the microtubular spindle fibers. So it is observed by that the centromeres, the center where the chromatids are attached, they split, which leads to separation of the chromatids of a single chromosome. Then after separation, the chromatids start to move toward the opposite pole. So next phase is the telophase. What happens in the telophase? At the beginning, as you know, it is the last phase, it is just reverse of the prophase. What happens in this phase? At the beginning of the final stage of mitosis, that is the telophase, the chromosome reach their respective, means they move to the opposite pole, and there they start to decondense. Decondense means, as you know, the chromosome attain the shape of chromosome at the metaphasic stage. Before this, the chromosome is present in the form of chromatid. Same happens there. The chromosome which get separated during the anaphase, they move toward the opposite pole, and at both the poles they get converted into the chromatin. So the key points of this stage are that chromosome, they get cluster in the form of chromatin at the opposite spindle poles. So they lost their identity as a single chromosome. Last, as you know, it is just opposite of that of the prophase. So in this phase, the nuclear envelope, it appears again, and the organelles like nucleolus, Golgi complex and endoplasmic reticulum, they start to appear again. So the four phases are prophase, metaphase, anaphase and telophase, in which the division of the nuclei takes place. So all these phases are considered as karyokinesis. Karyo means nucleus, kinesis means movement or the separation. So next is the cytokinesis. So what is cytokinesis? Cyto means cell or cytoplasm, kinesis means division. So mitosis is accomplished not only by separation of duplicated chromosome. It is not only that chromosome gets separated, that is only karyokinesis. It is necessary that with the division of nuclei, the division of cytoplasm also takes place. So the division or the formation of daughter cell get completed only after the cytokinesis, and after cytokinesis one cell get converted into the two. So in animal cell, how the cytokinesis takes place? In animal cell, the cytokinesis, it appears with the appearance of a furrow in the plasma membrane, and this furrow when deepens, means it deep inside, furrow is in this form. When it deep inside, it leads to the separation of the cytoplasm around each of the DNA containing nucleus. So next, in the this phase of nucleus, the DNA is formed in the form of the chromatin. Next, the important point is about the plant cell. As you know, animal cell lack the cell wall, so there only separation of plasma membrane or the division of plasma membrane takes place, while in the plant cell around the plasma membrane cell wall is present. So necessary point here is the formation of the cell wall. So the cell wall formation, it start at the center and it grows outward, means it is not like that of plasma membrane which start from periphery and end up to the center. It is just opposite of the plasma membrane. It starts from center and moves towards the periphery. So the formation of the new cell wall, it begins with the formation of a simple precursor that is said to be the cell plate, which represents the middle lamella. Then it is present between the two cells. So what happens, as you know, for the formation of cell wall, the necessary component which plays an important role is Golgi complex. So what did Golgi complex do? It accumulates the number of vesicles at the center of the cell, which leads to formation of the middle lamella, and finally the deposition of the adjacent membranes takes place and two cells are formed. Next, at the cytoplasmic division, what happens? The organelles like mitochondria, the plastids, these are divided equally to both the cells. And as you know, it is not always necessary that after every nuclear division the cytoplasmic division takes place. It is not always necessary. Sometimes the karokinesis occur on repeat, while the cytoplasmic division do not takes place. That kind of stage is said to be the syncytium. Syncytium is the state in which number of nuclear divisions takes place without any division of cytoplasm, and this kind of syncytium, it formed or it result in formation of the liquid endosperm in the coconut. It is a nuclear type, as we have done, and we will do this in next chapter. Next is, what is the significance of mitosis? So mitosis is a very necessary kind of division for our cell. As you know, all the somatic cell or all the organism which are undergoing asexual type of reproduction, all the lower plants and some special social insects which are usually haploid, means they have haploid DNA content, all these organisms divide by mitosis. So for the survival and the growth of the organism, mitosis is very important type of division. Mitosis usually it results in production of diploid daughter cells. If the, as you know na, after mitosis DNA content remain the same. If the cell is 2n means the diploid, then the identical genetic material will move to the progeny, that the cell will be diploid. And for our body growth from zygote to adult baby, and from baby to adult, and in adult also, our cells are dividing daily. Even our cells are dividing at every second or minute. The necessary for that is the type of division that is said to be the mitosis. So as you know, it is not only necessary that mitosis occurs without any signal. This is not the case. Mitosis requires special type of signal in which the nucleocytoplasmic ratio it get disturbed. It divides, the cell divide only when the nucleocytoplasmic ratio it undergoes imbalance. So for mitosis, it is necessary that if you have any kind of injury, to replace that injury, to replace those dead cell, the necessary point is the division of the cell. So for that division, cell must grow first in size, then it will undergo the division. Mitotic division also occur in the meristematic type of tissue like the apical meristem, lateral meristem, means every cell which is undergoing continuous division. The result of those division, it depends only on a single division, that is said to be the mitosis. So that's all for mitosis. It is very important division for the somatic cell division and for the organism which are asexually reproducing, because they can grow only if the mitosis takes place. Thank you.